Hello, my name is Victoria and welcome to this short presentation on the nervous system and skeletal muscle. In this week's lecture, we will look at the importance of chloride ions in the central nervous system and skeletal muscle, and we will do this by discussing the following below. To begin with, GABA, or gamma-aminobutyric acid, is the major neuroinhibitory transmitter in the central nervous system. It is found in high concentrations in the cortex and limbic system. It is inhibitory, meaning that it decreases or inhibits the neuron's ability to generate an action potential. GABA influences neurons via a large number of receptor subtypes, which are grouped within three major classes. These are GABA A, GABA B, and GABA C. GABA A and GABA C receptors are linked to GABA-gated chloride ion channels, causing inhibition of neuronal firing. GABA B receptors are associated with potassium and/or calcium channels through G-protein coupled receptors. Most of this presentation, however, will focus on GABA A receptors. So let's quickly review how an action potential is generated. As we know, the resting membrane potential of the cell is minus 70 millivolts and is primarily determined by the potassium concentration gradient and the cell's resting permeability to potassium, sodium, and chloride ions. A change in either potassium concentration gradient or permeability to ions changes the membrane potential. For example, an increase in sodium permeability into the cell down its electrochemical gradient will depolarize the cell, ultimately reaching threshold and creating an action potential. GABA, however, inhibits this process of producing an action potential through hyperpolarization, and we will discuss this in more detail now. GABA is packed in vesicles and stored in the postsynaptic terminal. They stay there until the arrival of an action potential causes these vesicles to release their contents into the synaptic cleft. When GABA is released, it binds to a specific receptor on the postsynaptic membrane. This binding opens specific ligand-gated ion channels that are selectively permeable to chloride. The opening of these channels allows the negatively charged chloride ions to enter the cell and makes the resting potential of the postsynaptic membrane more negative. This leads to hyperpolarization of the postsynaptic membrane and is called an inhibitory postsynaptic potential, or IPSP. It is inhibitory because it reduces the probability that the postsynaptic cell will reach threshold and fire an action potential. GABA is important as it helps to modulate and maintain different functions in the nervous system, including sensory and motor processing, central autonomic control, sleep wakefulness, emotions, and cognition. It is also typically known for its calming effect and regulation of anxiety. Clinically, however, the main relevance of GABA A receptors is that they are specifically targeted by benzodiazepines, which we will discuss in the following slide. Benzodiazepines are a class of drug commonly known as minor tranquilizers and sleeping pills. They are typically prescribed for anxiety, insomnia, muscle relaxation, and epilepsy. Benzodiazepines reduce the excitability of neurons through their strong affiliation with GABA. Benzodiazepines have a high affinity with GABA and act as a modulator on the GABA A receptor. The GABA A receptor complex has a particular binding site to which the BZD attaches to, and you can see it in this diagram here. When the BZD binds to the site, it stimulates the chloride channels on the GABA A receptor to open, hyperpolarizing the cell and creating the inhibitory effect through the central nervous system. Benzodiazepines therefore act on GABA to calm the nervous system. Valium, or diazepam, is one of the most common BZDs used for anxiety. However, it has the potential to interact with alcohol and other barbiturates, increasing the effect of the medication, for example, oversedation and memory impairment. Alcohol alters the effects of the medication without changing the medication's concentration in the blood. And with some medications, such as barbiturates and benzodiazepines, alcohol acts on the same molecules inside or on the surface of the cell as does the medication. And these interactions may be synergistic, That is, the effects of the combined medications exceed the sum of the effects of the individual medications. This can lead to substantial impairment within the central nervous system and may place an individual at risk of an accidental overdose. Now we will move on to the role of chloride ions in skeletal muscle. The CLC1 chloride ion channel is responsible for chloride conductance in skeletal muscle. These are voltage-gated channels and involve a common and fast gate mechanism. The channels are located along the sarcolemma and within the T-tubule of skeletal muscles, which you can see in this diagram in the orange. The opening of these channels typically increases with membrane depolarization. Chloride accounts for 2/3 of muscle membrane conductance. And chloride conductance is important as it contributes significantly to repolarization of action potentials and helps to maintain resting membrane potential of the skeletal muscle. Skeletal muscle channelopathies are caused by alterations of the electrical excitability of the skeletal muscle fiber membrane. Myotonia congenita is the most common skeletal muscle channelopathy and is caused by mutation in the CLCN1 gene, responsible for the main skeletal muscle chloride channel. The mutation causes a large shift in the gating potential and prevents the chloride channels from opening when required in repolarization. Reduction of chloride conductance results in myotonia. The myotonia is essentially an increase in muscular excitability and contractility with decreased power of relaxation. So that concludes today's presentation about the importance of chloride ions in the central nervous system and skeletal muscle. Thank you for listening.